Thank you very much. And today I'll talk about Metabolomics in Tohoku Medical Megabank Cohort project. The Institute Tohoku Medical Megabank organization was founded based on national project Tohoku Medical Megabank project. This project aims creative reconstruction of Tohoku region, Japan, which suffered from the Great East Japan earthquake about 10 years ago. And for support of residents in tsunami-suffered area, support of community medicine of the damaged area by establishing next-generation medical systems, and for recruitment of medical professionals to the suffered area, we started this project. And in this project, we started and did biobank in order to develop personalized healthcare systems. So in this project, we conducting two types of prospective cohort study. One is a community-based cohort study, which recruit residents from a coastal area in two prefectures suffered from earthquakes. The other is a birth and three-generation cohort study, which recruited three generations, offspring, parental and grandparental generations. So from 2013, we started baseline survey, and at the end, 2017, we ended baseline survey and recruited more than 150,000 participants. And now we are doing first repeat survey, and from this year, we will start a second repeat survey. In this cohort project, we not just made a biobank, but integrated biobank. Tomo set up this integrated biobank and analytical center execute standard analysis of sample because rapid depletion of samples. So we distribute not only sample, but analytical information, analysis information. So we collecting many kinds of samples, blood, urine, genomic DNA, and based on these samples, we are doing many kinds of genome and omics research. In addition, we also collecting many kinds of informations such as lifestyles, including foods based on questionnaires, psychological condition, experience of disaster. And in addition, was a part of participants, we also collected MRI informations and physiological examinations and psychological assessment. So based on these collected information and samples, we integrated biobank. And in the case of information, we stored to our original database, Tohoku Medical Megabank integrated database called DBTMM, and distribute this data to scientists in academia and industry. Next session, I'll talk about Omics research. In our Omics research project, we mainly focus on a metabolome analysis because metabolome is influenced by both genome and environmental factors. In addition, it's influenced by a microbiome and other factors. And furthermore, also metabolome influence other omics layers, genome, transcriptome, and proteome. So we mainly focus on metabolome research. So the role of our metabolome is basically two aims. One is a classification of individuals by molecular phenotypes. The other is survey of phenotypes change over time. Based on this research, biomarkers for disease and healthcare, and make methods for estimation of risks for disease onset based on analytical data. So for omics research project, we firstly developed our omics research platform, mainly based on mass spectrometry and nuclear magnetic resonance spectrometry. So in the case of mass spectrometry, we developed several methods during our cohort project. At first, we started research based on non-target type analysis. And from then, next, we also started target analysis based on gas chromatography-based methods. And recently, from 2018, we also started target analysis based on Biocrates absolute kits. And recently, we moved to next system, MxP Quant 500 kit. Based on this data, we are developing Japanese metabolic reference panel and other association study, which talked next sections. So in the case of MxP Quant, our NMR, and we found good correlation between 500 kit and data. So at the end of the year, we until last year, we collected data for approximately 4,000 participants, and we released this data our database Genome. This is an example of data, and difference of metabolome depend on sex and age. These differences are observed for each metabolite. So this next session, I'll talk about our omics research based on our open database, Japanese Multi-Omics Reference Panel, JMOP. So this database is developed on our organization, and this database is open integrated database, and including 25,000 metabolic data and whole genome sequence data. Much omics layer included such as genome, metabolomics, transcriptome, proteome, metabolome, and recently metagenome was included. In the case of metabolome, we distributing statistical data, and in the case of NMR, 5,000 samples metabolite concentration information, and in the case of mass spectrometry, targeted gas chromatography and targeted LC mass spectrometry, including Biocrates kit. And not only distribution information of each metabolite, but also we providing many other informations. So this is an interface of our omics research database, omics database, GMOP database. For each metabolite, you can see get information such as distribution in age dependency and correlation, and we also provide comparison information between baseline and repeat survey, and depend on pregnancy and depend on genetic variation. So this is an example of the information of correlation between metabolites. Each information is observed by clicking each icons. So if you click correlation icons, you can see these windows, and you can get information correlated metabolites, interested metabolite. So in the case of, we obtained association result between with genetic variants, so we also provide such information. We also provide information of metabolite change over time based on analytical result of baseline surveys result and repeat survey. So this is an example. For the left side, you can see a difference during 4 years glucose metabolite. And some people largely changed, but many people not so largely changed. And we colored each individual's point by age class. And as you see, glucose concentration slightly increased depend on age. And on the other hand, in the right slide, you can see difference of glycine during 4 years. Basically, glycine concentration is not changed during 4 years, but some people has larger concentrations. And we colored each point by each individuals by a specific genetic variants, genotypes. And as you see, glycine concentration is largely depend on genetic variants. So we are providing such information in our database. We also provide metabolite change depend on pregnancy. So many kinds of metabolite change during pregnancy. Distributing these statistical data in Genome, and we expect our database can be used for many kinds of researches, and such as case control studies, or identifying environmental factors, or verifying drug intakes, and other researches. So, final section, I'll talk about association study with genome and cohort data. As previously talked, we collecting many kinds of information in our cohort study, such as health survey items, questionnaires, including lifestyles and diet, and blood test items, and MRI data. And we also analyzed samples and obtained many kinds of genome and omics informations. And now we are investigating association study of these informations and metabolome. So from next section, I especially focus on association study with genome. So about 4 years ago, we released first result about the Metabolomics genome-wide association study. In this study, we used only 500 samples, but we identified 5 associations. So we expanded the data further MGWAS study. So recently, we released the next expanded version based on about 1,000 participants' metabolic information, including 200 metabolite information. And we found many associations in this study. And we also investigated the effect of rare variant, because in this MGWAS study, we used whole genome sequence data for 1,000 participants. So we investigated the effect of rare variants and found some rare variants affect metabolite levels much more compared to common variants. So in addition, we mapped these variants and common variants to the structure of the corresponding gene's product, because these rare variants is variants which change amino acid in the proteins. And as you see, compared to the common variant position, arginine 53, a variant with higher effects locate on near the catalytic center, which indicating these rare variants largely affect the catalytics. So we suggest the diversity of individual metabolic phenotypes is derived from the combination of common variants with moderate effect and rare variants with much more deleterious effects. In the case of the associated genes found in this MGWAS study, we also investigating previous association reports and found many genes previously reported to be associated with a variety of diseases. So our Metabolome genome-wide association study is useful for investigating such, also investigating for such diseases. So based on these results, we suggest Metabolome is very effective intermediate phenotypes. And compared to the traditional GWAS study, which directly investigates the association with genome and phenotypes, such as disease, Each effect of each genetic variant is smaller. So conventional GWAS study cannot detect associations even if large sample numbers. And on the other hand, metabolome is association with metabolome and genome is strongly associated. So we can investigate more effectively the effect of genetic variants to phenotypes. On the other hand, metabolome association and relation of phenotypes and metabolome intermediate phenotypes such as metabolome is very easily investigated by experimental verification compared to genome. So we think metabolome analysis is very informative for cohort in the cohort study. This today's summary. Omics research project developed the method of high throughput analysis of metabolome and proteome in plasma. And based on the analysis of more than 25,000 cohort samples, we provide standard information of plasma metabolome in Japanese population and provide data on database Genome. And we analyzed plasma metabolome of participants in the repeat assessment survey and identified the change of metabolome in the same individuals between baseline and repeat surveys. So finally, we investigated association between metabolites and genetic variants by MGWAS study and revealed that many kinds of metabolites are influenced by genetic variants. This is the acknowledgement. Thank you very much.